Hi everyone, welcome to my channel Pure Biology, and today's topic is tryptophan operon system, and I have already explained the lac operon system and lac mutation in my previous videos, so you please check out that videos and continue with me. And obviously I am very grateful to all of you who have already subscribed my channel, and the new ones, we are saying newcomers in my channel, I request you to subscribe this one for the growth of this channel. Okay, so today's topic is the tryptophan operon system. What is the tryptophan operon system? Like that, the lac operon system, the tryptophan operon system is another operon system that is seen generally in the E. coli. Okay, that is seen in E. coli. So this is another operon system. That what is the role of actually of that operon system? It generally enables the E. coli to manufacture any amino acid that is lacking in the medium. Okay, so E. coli remain a medium where for their growth and the reproduction another amino acid is very important. Okay, but there in the medium this amino acid is not present. So this operon system that is present in E. coli, it has the capacity to produce any kind of amino acid that is needed for the E. coli. Clear? So when an amino acid is present in medium, though the gene encoding the enzymes for biosynthetic pathway for that amino acid are turned off. So what is saying here? When any amino acid that is present in the medium, though the gene encoding the enzymes for the biosynthetic pathway for that amino acid are turned off. So what is saying that if any amino acid that is very important, for the growth of the E. coli obviously needed in the medium, because without that amino acid their total transcription translation process or the cell growth system that may be stopped. Okay, so that's why this operon system is very important. In this case, the gene activity is repressed. I am saying that the gene activity is repressed, and when a chemical, and here if the chemical is added to the medium, the gene organization of the tryptophan biosynthesis, this is another part. Okay, so in this case, gene activity is repressed when a chemical is added to the medium. So here, what is saying? Please clearly understand. In the lac operon system, we have seen that if one chemical is sometimes added to the medium, then the transcription process become continuing. But in this case, this is not possible, that any substance or chemical, if we add it just within the medium, then the total gene activity or the translation process totally stopped. So here the any chemical cannot function. Okay, but in case of what we just seen in the lac operon system, in that case chemical lactose that was very important for the medium, when we added in the medium, then the process become continuing. But this is not possible here. So this is totally another different types of process from the lac operon system. Okay, that's why this system is very important for the E. coli, because we cannot add any chemical within the medium. So E. coli must have one kind of system that try to produce this type of amino acid that is already absent in the medium, and which is very important for the growth and the reproduction of the E. coli. Okay, so this is the actual role of the tryptophan operon system, because with particular amino acid is not present, that means total growth and processing or the cell growth, you can say cell growth of the E. coli, that becomes stopped. Okay, so now we have to learn actually in the tryptophan operon system what are the major parts that are related for the tryptophan operon system. So you just think this is the rough sketch of the tryptophan operon system medium's structure. So what is that? This is the 3R gene. This is the position of the 3R gene. R means regulator. 3R. This is the TRYP promoter. And within the promoter, this is the O operator. And this one is the T and L. A for leader. And within the leader, there is another place that is called the attenuator. Okay? And or at site. A-T-T site. At site. Okay? This is at site. Next, these are the structural genes obviously. TRYP-E, TRYP-D, this is TRYP-C, TRYP-B and the TRYP-A. All the tryptophan genes, the structural genes. Okay. So, what we have to learn that the 5 genes are there in this system, and the promoter operator are upstream from the TRYP gene. You can see that this is the upstream and this is the downstream position. So this is if this is TRYP-E, so the promoter and the operator and the regulator that are already present the upstream portion. Okay, between the promoter and the operator region and TRYP-E is a short region called the TRYP-L. This is, you can see, this is the TRYP-L and is situated just between the TRYP-P and the TRYP-E. Okay. Obviously, operator is included within the TRYP-P promoter. And within the TRYP-L, close to the TRYP-E is an attenuator site, plays an important role in the regulation of the TRYPTOPHAN. Okay. And this is the at site. I have already said that the attenuator or the at site, it had obviously another role in the regulation of the TRYPTOPHAN system. Now we have to learn, sorry for the disturbance, okay, sorry. Okay, so now we have to learn how the expression of the gene is seen in presence of the tryptophan. The product of regulator gene is an aporepressor protein which is inactive repressor and that alone cannot bind to the operator. So what is the actual thing is happening here? You can see here, this is the TRYP-R regulator, this is the TRYP-P promoter and these are the genes. So in such a situation, the TRYP regulator, it can produce its product, that is a repressor protein. But this repressor protein is very inactive and it has no role for stopping this transcription process. Clear? Just clearly understand it. Its capability is to produce the protein, repressor protein, but this repressor protein is inactive and that's why it cannot bind the operator site to stop the total structural genes function. Clear? So, what happened is the tryptophan is, and that's why it is called the aporepressor protein. This is called the aporepressor protein. That's why it has, you can say it is totally inactive protein. Clear? But when the tryptophan is present in the medium, clear? The tryptophan is present in the medium, it interacts with the aporepressor. Okay, it interacts with the aporepressor and aporepressor that becomes in that situation becomes active. Okay, and it has the capability to bind with the operator. Okay, it has the capability to bind with the operator system, and as the repressor protein that already remain in the aporepressor but after binding with tryptophan that becomes in the active situation, and when it binds to the operator, the total transcription process of this gene that becomes stopped. Okay, and so what happened? The tryptophan, the tryptophan protein coding genes by the RNA pol. So, and it generally stop the function of the RNA polymerase. Clear? Stop the RNA polymerase, and so the transcription process of this gene totally stopped. And here the true process you can see that is, if the severe amount of tryptophan is present in the medium, then the genes, 3 genes are expressed maximally. Clear? The 3 genes are expressed maximally. And under the less severe condition, that means tryptophan is present in the medium but less, then what happens? The 3 genes are expressed less maximally. Okay, so this is actually the tryptophan operon system. It is very easy, and you can obviously after sorry for the disturbance. what is saying here, the expression in presence of the low concentration of the tryptophan, that means under the severe tryptophan starvation, that means the TRYP genes are expressed maximally, and if the low concentration is there, that means the genes are expressed less maximally. So okay, after that we have to learn what is the actual attenuator very briefly, because the attenuator I will explain it properly as another video obviously. So here its role is just briefly I try to describe you. Okay. So here the short, what is the actual function of the attenuator, we just learned very shortly. The short transcripts are terminated by the attenuation process. Actually the short transcripts that are terminated by the attenuation process at the site of the act site or the attenuated site. And the ratio of the transcripts that include the structural genes inversely related to the amount of the tryptophan. Okay, so actually the ratio of the transcripts that include the structural genes, that is inversely related with the presence of amount of the tryptophan in the medium. And if the more tryptophan is present in the medium, then greater is the proportion of the short transcript. Okay, greater is the proportion of the short transcript. So this is the actual tryptophan operon system. Okay, and I will explain another topic in my next video.